So let me first say that my wife and my parents are in the audience, so if it's really good, it's all because of them, and if it's really bad, it's all my mother's fault. No, I'm just kidding. Just kidding. No, it's a joke. Anyway, so thank you for coming, Mom and Dad, and Jill. And anyone else, all of you. I actually think it's amazing that people, so many people come to these lectures, and it's really just a wonderful thing about our community. So I was thinking about how to give this talk and sort of where to pitch it, and I thought that I could give a sort of overview summary of where it is and talk about things, or I could try and actually speak really very current, like if you went to a scientific meeting, this month, what would you hear about this topic? And I decided to do the latter, with the challenge being that I'm going to try and do it in simple English. I'm going to try not to assume too much about what people know. And if I miss and say things that you feel like you have no idea what I'm talking about, feel free to actually shout out and say, can you explain what that is? Because my goal is to be understood by anyone, but I figured rather than sort of summarize 5 years ago, I'd just sort of say where we are today. So the topic that I'm going to talk about is type 2 diabetes. That's the most common form of diabetes, and it's really remarkable how many people, and really horrifying how many people have this disease. It's estimated there's something like 300 million people who have it. It is a leading cause of disease of the blood vessels, so leading cause of heart attack, of a kidney disease, of blindness, of amputation. And I don't want to sort of create any concern among people who have this disease, which I'm sure people in the audience do, because it's as much as 10% of America have this disease. But our current forms of therapy, both dietary and drugs, fail to arrest the disease, let alone reverse it. So if you sort of think about it, you'd like to live in a world where you get a disease and then there's something you can take and actually it just gets better. This is a disease where actually we can't even stop its progression, let alone reverse it. And it's a disease, it's often thought of as being a disease of the developed world, and that was once the case. But actually this slide, which is from the International Diabetes Federation, shows that the vast number of people, the numbers here are the millions of people with the disease today, the number projected in 2030, and the percentage growth. And you can see that, for example, in Africa, today there are 12 million people with type 2 diabetes, estimated to grow to 24 million. 100 million people in India are estimated to have this disease in 15 years or so. So it's truly a worldwide epidemic. And if you read in the newspaper, you'll often see this is the statement that this is a disease of lifestyle, and in some ways it is, because the environment in which we live today, with food being readily available, highly palatable and inexpensive, relatively speaking, and many people's jobs not requiring physical labor, raises the likelihood of this disease in a susceptible individual. But you'll also read then a statement which I think is not true, which is that because it is a disease of the environment in which we live, that all you need to do to stop it is change your personal lifestyle. You go on a diet and you exercise. This often cites a study called the Diabetes Prevention Program as the source of this information. The Diabetes Prevention Program is a landmark study. It was led by David Nathan, who was my clinical mentor, along with a man named John Godine at Mass General Hospital when I was training in diabetes, and David conceived of and led this study. And what this study did was it randomized 100,000 people to find 5,000 people who didn't have diabetes type 2, but were at high risk of it because they were overweight, or they had a family history, or they'd had diabetes during pregnancy. And then they randomized these people to either standard of care today, which is still the standard of care today, which is nothing. So we don't actually identify people in this high-risk category, nor do we do anything other than say, you should live a healthy lifestyle. That's still the truth today. They then randomized these people, either that placebo arm, standard of care, or to a pill, metformin, or to an intensive lifestyle intervention, where people are given a personal dietary coach, a personal exercise coach, a fitness, et cetera, and they followed them now for 10 years. And what you can see is that the study was strikingly positive in that if you were in the placebo group, that is the standard of care today, there was a shocking level of type 2 diabetes in these people, 50% over a 10-year period. 50% of these people went on to get type 2 diabetes. And the study was strikingly positive because if you were in particular, the lifestyle arm, which was actually more effective than the current standard of therapy pharmacologically, which is metformin, you would reduce that by a very statistically significant and real amount, which is to reduce it from 50 to 40%. Or another way of looking at it, if you look at how these curves are essentially completely parallel, but simply staggered, what it's done is it's delayed, if you look, about 2 tick marks. So you get about 2 years of delay. That's the best we have today for this disease. It's quite shocking, actually. And if you're surprised by that, just note that the disease is exploding in prevalence. So if we had really effective prevention and therapy, we could say it's because no one's willing to change their lifestyle, but at least my experience clinically was people with this disease often are very motivated. They try their best. It turns out it's very difficult to conquer this disease. So I take away from this that actually it's not sufficient, although it is necessary, to say that we should change our lifestyle and use standard of care. We actually need something better for this disease because it's exploding, we don't have great therapy, and it's still today a major, major problem. So where do new treatments come from? And this is actually, in some sense, the motivation for my talk. I'm actually going to focus now, I'm going to step aside for a second and talk not about diabetes, but the development of new therapies. This conversation is, by the way, going to get depressing for a couple of years, and then we're going to have some hope, okay? But I want to be honest about the problem. So this is sort of an amazing slide, okay? This is now not diabetes, all of the pharmaceutical industry from 1970s through to the present. The curves here are, this here is the total spending on research and development of pharmaceutical companies. And that goes from very low, in 1970, up to on the order of $50 billion a year. Okay? So that's the investment. That's not just, part of that is because the pharmaceutical industry succeeded and its revenues grow. But actually the green line is the percentage of all revenues, which are spent on research and development, and it goes from something like 10% to something like 16 or 17%. So in this period, the reason this curve explodes so much is not only the pharmaceutical industry succeeding in developing drugs and growing to be a very big industry, but it's actually doubling its percentage of revenues it spends on R&D. The red line is the number of drugs that are approved each year. And you will note it does not go up. Okay? This year, actually, this past year was a good year. There were more drugs approved this past year than in some previous years. But nonetheless, this is sort of a shocking lack of productivity despite a phenomenal investment. And why is that? Well, it turns out that the single most important number that you need to know is like 2 to 5%, call it 3% if you want. What is that number? That's the number, that's the percentage of drugs that enter human clinical trials, drug candidates, that emerge the other end as a safe and approved therapy, 3%. So if anyone ever said that, so actually how much does it cost to develop a new drug? Turns out all you need to know is this slide, by the way, to figure, there are some people who like to build this thing bottom up and they calculate all the costs to develop a new drug. All I do, and I'm not that smart about this, I say, well, it's $50 billion a year and there were like 25 drugs, right? So that's $2 billion a year per drug. But where does the money go? Well, it turns out it's not in research, okay? So it's not actually, sorry, what was that? So the comment was it goes to the fat cats in the pharmaceutical industry. I won't really take that one on, but what I will say is it's good to have therapies that actually work, and actually this is how they're developed. And so if there's an alternative model, we have yet to see it actually. So it's good to have therapies that work at least, and we do have a lot of therapies that work that have come from this process. The real problem, I would argue, or at least the problem I'm going to focus on, okay, is the lack of productivity, okay, not the salaries of the individuals. And the lack of productivity is because almost everything we put into patients as a candidate drug actually fails. It doesn't emerge. And so if you figure that there's 19 failures for every one success, and each one of those fails often late in the process, and you have to spend tens and hundreds of millions of dollars on the clinical trials, and you multiply that by 20, that's not because people are taking too much cream off the top. That's not because they're ill-intended. That's because there's something wrong with the way we're doing it, okay? And if we could actually increase that percentage from 5% to 10%, or 15%, which is what it was 20 years ago, okay, then actually all of a sudden it would cost a lot less to develop drugs and the whole system would work much better. Why do they fail? So if you ask a group like this, why do they fail, or a group of scientists, a group of doctors, they will often say, oh, well, they must fail because of toxicities. And actually, 25 years ago, that was true, that the reason they failed was often because they caused toxicities. But actually, the pharmaceutical industry and chemists have become quite good at what's called preclinical toxicity prediction, figuring out which drugs will be toxic, and that's much smaller percentage of failures now. The single largest failure mode, which is depicted in these two summaries that were published, 3 summaries actually, of failures from the pharmaceutical industry, is lack of efficacy. They simply don't work, okay? They're put into patients. They all work, by the way, in preclinical models. No drug goes into a person for testing unless it is proven both safe and effective in an animal model. And yet they fail 19 of 20 times in a person. So why is that? Well, again, it's in part because human biology is incredibly complicated. It's very difficult to do experiments, and we don't understand it that well, right? So that's not actually like, again, someone's fault, like someone's doing something wrong. It's simply that we don't actually understand human beings very well, and it's understandable why we might not, because you can't, like a fruit fly, you can do experiments in a laboratory, so you can learn a lot. But people, you can't, for all sorts of good reasons. But when you think about why it's hard to develop drugs, just realize that the fundamental problem is not people who are ill intent or people who aren't smart or aren't trying. It's because we don't understand human biology well enough to engineer the system. And that's what you're trying to do when you're trying to develop a drug. You're trying to engineer a solution that will fix a problem that you don't understand, okay? And when we do understand it, then actually you have a much better shot on goal. So I would argue that the reason for the most part that drug candidates fail, it's not the only reason, this very complex problem, is not actually many things, but it's actually just that we actually don't have a correct therapeutic hypothesis. In other words, we don't really know what to do to fix the disease, and the only way that we actually can figure that out is by developing an agent with the best, a drug with the best evidence we have, and then testing it in humans and seeing if it works, and that doesn't happen very often. So how do we get out of this? Well, in order to understand how we might improve this, we have to understand how is it done today. And the way that it's done today, and this is not how it was done 25 years ago, okay? When one could argue it was more productive. The way it's done today is that we have so much tremendous biology, and in particular, we can learn so much from very tractable experimental systems that we develop a picture of how the system works, often from cell and animal models, and then we decide that there is a target. And the target is some constituent of the cell that you're going to develop a drug against, and you have to decide whether you're going to inhibit it or activate it. And then you, having done that, so you find a chemical, and then you test it in the best, you can't go straight to people, and no one would suggest you could. You have to test it in models. And these things are often called validated targets and validated animal models. And that sounds very good, but we know that most of the time when you go into the patient, it doesn't work. And again, understand that the people I work with and have gotten to know the pharmaceutical industry are tremendously smart, well-intentioned, hardworking people who don't do things that are silly. So like they don't go into a clinical trial unless all the boxes that you could check, that you could think of and that I could think of beforehand were checked as to hopefully the best we could know that this was likely to work, and yet it doesn't work that often. So I think renaming this, what we really have to do is realize that in this approach, what's really happening, it sounds good to say validated target, validated animal model. What it really is you're stating a therapeutic hypothesis back here. If I were to inhibit this, it might help a patient. And then it takes 10 years and like $100 million to get from the other side and figure out if you're right, okay? So what we'd really like to do is front load this process. Figure out in advance, as best you could, whether or not perturbing a given target with a drug, making it more or less active with a drug, would, in the context of patients, not of models in a lab, would actually benefit that patient in the exact way you would like without toxicity that's due to the actual target perturbation. You'll never be able to rule out perfectly that there's not some other effect of the drug, but at least the one you intended you'd like not to cause a problem. And that you would do this rather than extrapolate from the laboratory and test that hypothesis for the first time in patients by doing the clinical trial, because that takes 10 years and costs $100 million. Okay? So that to me is why I got involved, or I can't say this is why I got involved, because actually there's been an evolving thought process, but if you ask me, and because of where I work and the kind of work I've done, people say, well, was the Human Genome Project worth it? Did we get much from it? And that's often framed in terms of, can we someday sequence someone's genome and predict what will happen to them? And I say, that's a whole other lecture, I'd be happy to talk about it, but it's not really where the value is. The value is being able to understand in patients how the disease works so that we can do a better job of actually trying to intervene. And the genome, it turns out, is a tremendously powerful tool for doing that, for trying to understand. Okay, so I wrote an article a couple of years ago, a year or so ago, with these two people, Robert Plenge and Ed Skolnick. Robert Plenge was at the Harvard and the Brigham, but now is actually at Merck. Ed Skolnick is the legendary president of Merck Research Labs who developed many of the breakthrough drugs that we have today, including statins, which lower cholesterol, HIV drugs that made HIV a chronic disease, vaccines, et cetera. And we wrote this article about the idea of using human genetics as a way to validate drug targets. So in other words, using the patient as the way to do it as opposed to some other approach. And the simple picture is that a drug you want has the following properties. So in this slide, the y-axis is just like a response that you'd like to see happen, and or a response in the patient. And here it's sort of the one you want to see in green, like that's the positive response. And then there's the one that's not so good, like a toxicity. Because almost everything, if you pushed hard enough, will cause a problem, on almost, not all, but most targets. And then this is sort of how much you've modulated the target. See that as like a dial, okay? And what you want in a good drug is that you dial it up from low to high, you get a beneficial effect before you start picking up a toxic effect, and you find the dose that sort of maximizes the benefit, doesn't have that risk. So that's sort of what a successful drug looks like. It turns out that a lot of the early successes of the pharmaceutical industry, not all, but many, were based on following experiments of nature, often involving hormones. I'm an endocrinologist, a doctor, or trained as an endocrinologist who took care of patients with hormonal disorders. And what hormones are naturally occurring chemicals or proteins that float around the blood and have effects. And because the way that endocrine systems work, or at least the ones we know about work, is because there often was a syndrome where you made too much of it. There was a gland. My grandmother would have said, I'm a gland man. And she would have actually said that. So there's a gland, and it secretes the hormone, and some rare people have too much of it. So you learn what's the effect of dialing it all the way up. And then sometimes surgically or due to an autoimmune disease, you take out the gland, and you find out what there's too little. And then it turned out steroids, so probably many people have taken prednisone or know someone who's taken prednisone for inflammation and other things. Well, it turns out, how were they developed as drugs? Okay, well, it turns out there's a hormone called cortisol, and there's a disease called Cushing's disease where you had too much of it. And there's also in pregnancy, it goes way up. And there's a disease called Addison's disease where you have too little of it. And when it was figured out that that was due to cortisol, people realized that rheumatoid arthritis, and there was a Nobel Prize for this work, that rheumatoid arthritis would get better in people who had Cushing's disease, and it would go away. And they also knew what the side effects were, because they get diabetes and obesity and depression and things like that. And they also knew what happened if you withdrew the hormone abruptly, because you have your Cushing's disease, you'd have surgery to take out the gland that made too much cortisol, and they knew what all the side effects of withdrawal were. That's all before anyone tried to develop a drug. All right? So then, when they went to synthesize a chemical version of cortisol that became prednisone, or all the other drugs in that family, they knew what was going to happen. There was not that risk that it wouldn't work, and there was knowledge of what the side effects were, because they already knew from a human experiment of nature. So that was sort of, and it turns out like 30 or 40% of all the drugs that are used are endocrine drugs. They're either the hormone themselves, insulin, if you think about lots of thyroid hormone, these are just chemical versions or man-made versions of naturally occurring hormones, or they're things that block or activate the receptor, the key in the lock. And those drugs were developed without much risk that they wouldn't do what we thought they'd do, because everyone already knew how the human biology worked, okay? And so the question then is, can you do that outside of hormones? And it turns out that human genetics has the property that sometimes people inherit a version of a gene that naturally dials up or down a system. And you can learn, and so for example in cholesterol, specifically LDL cholesterol, it turns out there's lots of genes that have been found that either give you too much LDL or too little LDL. This is actually the origin of the statin drugs that are used by millions and millions of people and have saved many, many lives. And those drugs, if you ask Ed Skolnick, who actually developed the statins, or if you ask Brown and Goldstein, who won the Nobel Prize and who harangued Merck to develop the statins, they already knew what happened in people when you had too much. They knew what the biology was, and this actually gave them confidence to go ahead when that project was hard, this is 30 years ago, to develop the statins because they knew something about the human biology. And at that time, there was a lot of arguing about, is it the case that LDL would actually be causing the disease or not? And a lot of people invoked a lot of different kinds of evidence, but Brown, Goldstein, Skolnick, and others said these genetic syndromes tell us that it's a cause, because they knew that when nothing else happened but this gene that raised the LDL, and it was actually in the receptor itself, the LDL, that could cause the disease. So they were confident if you could reverse that would, at least in those cases, and it turned out to work in almost everyone, to lower the risk. So if we're going to do that, that's the best case, where the genetics can sort of tell you what that dose-response curve is in advance. It's just like a hormone, a gland making too much and too little of a hormone, but it's a gene you inherited that made you have a lot or a little of something, and that guides you as to what you'd want to do with the drug. What you really don't want to do, okay, is you don't want to develop a drug where in advance you could know that modulating the target has no effect. right? But I can tell you there's lots of that going on. And I'll give you an example. Well, not intentionally, okay? So I'll give you an example if I have my slides right. Yeah, so how many of you have had your HDL cholesterol measured? Okay, some of you don't know it, but you've also had your HDL cholesterol measured because everybody has their HDL cholesterol measured. If you've gone to a doctor, you've had your HDL. Whenever they measure your LDL, they measure your HDL. Okay, so how many of you believe, so HDL, in fact, is, so LDL, this is data that shows that this is the LDL cholesterol level. And if it goes up epidemiologically, this is like taking people, they're walking down the street, you draw their blood, you measure their LDL, you follow them for many years and figure out which ones had heart attacks. The Framingham Heart Study was the first study to do this. And you find a linear relationship that the higher your LDL, the higher your risk of heart attack, okay? And there's this inverse relationship where the higher your HDL, the lower your risk of heart attack. And this led this to be called the bad cholesterol, and this to be colloquially known as the good cholesterol. And it also led to the implication that if you would lower this, that would be good. And if you could raise this, that would be good. How many of you have ever, actually, so I want to assure you, lest I commit a public health crime, that LDL, by every measure we know of, is in fact a bad cholesterol. By which I mean every genetic variant that has ever been found that modulates LDL cholesterol has the corresponding effect on heart attack. In other words, if it raises the LDL, it always raises the heart attack. If it lowers the LDL, it lowers the heart attack. And the statin drugs that lower LDL save lives, okay? And so I believe there are a few things in medicine that I am more certain of, okay, that lowering LDL lowers risk of heart attack and that's how statins work. You can read all sorts of noise in the literature about how statins do other things, but actually it's all confused, okay? Because the preponderance of the evidence when you integrate everything is lowering LDL lowers heart attack, and the effect of a statin on lowering LDL is exactly as predicted based on its effect on LDL. How many of you believe that if you were to raise your HDL, or actually, how many of you have ever been told by a doctor or the newspaper that if you were to raise your HDL, that it would benefit you? Okay, so I'm going to show, so the question then is, why do we think that's true? Well, it's correlated, right? But does correlation imply causality? Not necessarily. That actually is why, for decades, no one actually believed that you should lower LDL, because correlation did not imply causality, until the statins came along, and until the genetics told some people it was worth lowering it, but it was controversial. Then the statins came along and lowered it in 100 million people, and their heart attack rates all dropped. And then people became convinced it was true, and I'm convinced it's true. I think it's one of the premier things we really know in medicine, in terms of the evidence. And then everyone kind of, I think, gave HDL a pass, because H is only like a couple letters away from L, okay? I'm actually not kidding. I'm sort of kidding. So there is biology around how HDL works, but there was the only evidence that I believe really can convince you in people is evidence where you have some sort of randomization, you modulate the thing, and you see what happens, okay? And so what about HDL? I can tell you that billions of dollars with a B, okay, billions of dollars, that has gone to developing drugs to raise HDL, okay, billions. So how are we going to know? Well, the traditional way we know is we do a drug intervention. This RCT stands for randomized controlled trial. You have some people, you randomize them so that you know that you've taken out of the equation that the people who choose to do it are different than the people who choose not to do it. That's the problem with all of epidemiology that's behavioral and environmental, is the people who take hormone replacement therapy are different than people who don't take hormone replacement therapy. That's why it's a robust correlation. Taking hormone replacement therapy, patients have better outcomes, but in the randomized trials, it turns out they don't have better outcomes. So my view of it, although there's lots of noise around it, is people who, you can think for yourself about how people who take hormone replacement therapy might be different socioeconomically or otherwise than people who don't, okay? It's very hard to get rid of that confounding. But if you randomize, flip a coin, then you sort of take all that out, and you just say the people who got it or didn't, what happened? Okay? The trouble is you need to develop a drug and then do this, because how else do you do the intervention? You need a safe and effective way to do that test. Okay? So the idea is what's called Mendelian randomization, or sort of genetic randomization, is it turns out that a funny fact of biology is that when you make a sperm or an egg, okay, during the development of the sperm and egg, there's a randomization event. You have two copies, one you got from your mother, one you got from your father, and it's a coin flip, which one you pass on to the offspring. So if that individual, that mother or father, had two versions of the gene, let's say one that functioned normally and one that didn't function normally, and the not functioning normally copy cause the HDL to go up or down, then that's actually a randomization event. And if you do it all right, and there's lots of ways of doing things wrong, but it turns out in this case there's a way of doing it right, you can isolate that coin flip and then understand, with nothing else being different about the people except which version of the gene they got that modulated their anything you can find a gene version that modulates, what happened to the patients. So we set out, and we here is a group of hundreds of people, because to do the kind of study I'm going to talk about, you need actually lots of patients, and no one person can see and collect and organize that many patients, let alone bring together all the technology and analysis and methods you would need to do this work. And actually, in scientific audiences, when you show this, you always get a titter. You always get a little laugh, or sometimes a gasp of discomfort, or even sometimes horror, okay? And when I gave a lecture at, I won't say who it was, it doesn't really matter, but I gave a lecture at a prominent institution, and the person who introduced me, who's very supportive and lovely and a nice guy, said, David is going to give a lecture, and blah, 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 and he said, and he's the only person I know whose author lists are longer than the paper. It's more words in the author list than in the paper itself, which I pointed out, since the paper includes the author list, as a physical impossibility. But that's the kind of logic you get from people who aren't quantitative. But anyway, I'm just kidding. But the point is, our field used to publish papers. I tend to be tangential. Our field used to publish papers with few authors and no positive results. Now we collaborate, publish papers that bring all the data together, and we discover things. And amazingly, and just incidentally, some people in science are uncomfortable with that, because they actually think it's more important to have a short author list where you know that who did it and they did alone than to discover things. Or maybe they believe the things you discover when you work together aren't worth discovering. I don't know. But in any event, I just wanted to comment, because there was that titter gasp that I'm used to in the audience. But this actually project was really the brainchild of Ben Voigt, who was, and Seik Kathreson. Ben was, and Seik were postdocs in my lab at the time. Ben is now a professor at University of Pennsylvania. Seik is a professor at Mass General and the Broad. And the idea was simply to ask this HDL question, does HDL up or down affect heart attack, by using the genetics that Seik and others had learned in the previous years about genes that could change HDL and LDL, and ask what happens to the heart attack. And this study was particularly gratifying, I just want to note, because the thing we think is most important about the Broad Institute is the young people we can point to who had a dream, had an idea, have been able to actually achieve it because of the community and the capabilities. And I met Seik, again, another tangent, I met Seik when he was post-call at Mass General wearing scrubs. He asked to meet with me, he rolled in at like 9 in the morning, and he was very tired. He was like an intern. And he said, I want to understand which of the cardiac risk factors are affecting heart disease. Or I want to do that by using genetics. And it was 13 years later that he published his paper, and he's now the world leader in that field. And that was like an amazing thing that he did. That was his question, and he actually has become the world leader, and I'll show you some other of his work, but this is really his baby. So we gathered up the genetic variants that affect HDL, and then looked at them in like 100,000 people with and without a heart attack, and we measured their HDL effect. And as a control, we did the LDL. So we looked at other genetic variants that affect LDL, and I'm not going to show you the data, but every one of them had the predicted effect on heart attack in the exact same samples. So it's a controlled experiment where you say in the exact same human beings, you look at the genetic variants that are coin flips for LDL, and every time the LDL goes up, the heart attack rate goes up. Every time the LDL goes down, the heart attack goes down. What about HDL? And I'll show you just one example, which was, this is 116 data from 116,000 people with and without heart attack. Okay, so these are the kind of numbers you need to get to 99% power to have detected an effect if there is one. And I'll get back in a second to why we needed that. And this is a gene, it has the name LipG, it doesn't really matter. It's a gene involved in how HDL works, well understood biological role, and had a genetic variant in it, the position 396, some letter of the protein is different than another, and it's been shown that that genetic variant, which is known to change your HDL level, also changes the function of the protein in a known way and has no other effect, okay? It had absolutely no effect on heart attack rate. 0.99 to 1.01 was the change in heart attack rate. We actually did this for many HDL varying genes in this paper, and they had no effect on heart attack. Now, this might seem a purely academic interest, except during the period of time that we were doing this study, and unfortunately we were too late, there have been 4 failed trials of agents that raise HDL very substantially at a cost of billions of dollars, every one of which has failed. And some of which have actually caused toxicities without any benefit. And there's in fact a New England Journal, I believe, editorial just the other day about 2 failed HDL raising trials, maybe it's numbers 4 and 5, that cites this paper and cites all the failed studies and says maybe this really isn't the right hypothesis. I can tell you, you might ask, why did you go to 116,000 people? Did it really have to be 0.99 to 1.01? And the answer is, I'll let you ponder how enthusiastic you think the HDL field is about this result and other results like it. So this paper took 4 years to get published. We publish lots of papers in journals. We send it in and they say, that's a nice paper, and they publish it. This one was rejected from 4 journals. It was subject to, 20,000 patients not enough, you gotta go to 50, you go to 50, 75, you go to 75, you go to 100. And at some point, and actually took the failed clinical trial of torcetrapib, which dropped the value of Pfizer by whatever, tens of billions of dollars for the paper to actually be acceptable to some of the people. So I can't directly link those, but okay. So what the data from human genetics say, and also it's very consistent now with I think it's 5 failed clinical trials now, is that changing HDL may not be the good cholesterol. It's correlated with something good. I just want to not leave you confused. Remember, LDL, lower it. It's good to lower it. It actually benefits you. HDL is probably reflecting something we don't know, because it is very consistently the case that that higher HDL associated with something good. It's just not the HDL we're measuring, because there are ways we've shown and others have shown to change that HDL and nothing good happens. Nothing happens at all to heart attack. So probably a different way of modulating that system will work someday, but we don't know what it is. Okay. So to make such studies possible, that was sort of in some sense you can go home now, but that's sort of, except I've only killed things yet. I haven't actually come up with anything new. So I'll come up with something that actually gives you, yes. What was the control? So if by control, do you mean, what do you want to control for? Do you want to control that these samples really have heart attack and that they really could detect it?
People who did not get heart attack and have the same amount of HDL. Did you have that group?
Yes, so this is 116,000 people, half of whom had a heart attack or so, half of whom didn't, who have a whole wide range of HDLs and LDLs and things like that. And so we've isolated in this the variable of HDL in this particular figure for this one gene and done it for other ones. I'll be happy to talk more about it afterwards if you like. So in order to do that study, what you needed, it turns out, you needed to know the sequence of the genome of human beings, because that's where the DNA that is the genome, and you need to know how it varies, okay, like those variations in that genome. And you needed to be able to measure that stuff and relate it to diseases. And you needed to actually know which of those genetic variants actually affected diseases and traits. So there are lots of genes in that same set of samples that do affect heart attack rates. And there are other genes in that set of samples that affect LDL and HDL. And you can then ask which ones that affect this thing also affect that thing, begin to pull it apart. And before we really can do this work, we're gonna have to understand how these things all actually function. So I don't mean to suggest there's sort of the thing that's racing ahead over the last 30 years and accelerated over the last 10 is knowing the genome and how it varies, being able to measure that genetic variation in people and be able to relate it to which of the genetic variants affect disease. And that has become so powerful that it's raced far ahead of our ability to understand how it is that the genetic variants actually cause those diseases. Because what genetics is great at, it turns out, and the reason that I do it, is you can start with the actual patient in the actual state in which they live, the environment in which they live, and with knowing nothing about the disease, other than it has a genetic component, knowing nothing else with no hypothesis of how it works, you can find the gene, or genes. There's often many of them. And that's really valuable because you don't have any assumptions other than it's affected by inheritance. The bad news is you don't know anything about how that gene works or how you get from that gene to that disease. So then having found that, you got to go forward. And that's like early days for a lot of this stuff. But my colleagues and I here at the Broad and around the world, I started doing this work 17 years ago, and the first 10 years was largely about these first 2, and in the last 7 or 8 years, we've gotten very good at this, or the field has gotten good at this third one, and it's sort of still early days in figuring out for the new things we've found, how they actually work, okay? So what I want to do, I'm just looking at the time here, what I want to do is I want to jump to, just give me 1 second, I want to jump to something that now will hopefully be hopeful, okay? In terms of, because I don't want to suggest that all we've done is say we don't know much about disease, In many cases, not all, we don't have good therapies, and some things that we thought would work don't work, and we might be able to figure out how not to do those, although that actually would be helpful. If we could just eliminate the ones that don't make sense to do, then the success rate has to go up because you're doing fewer of the ones that fail. But nonetheless, you'd actually like to, and of course it wouldn't be very satisfying if that was all you could do. What you'd like to do is actually be able to find things that will work and nominate them. And so the idea here is going back to this experiment of nature concept. And what we want to do is not find genes that cause disease, but we actually want to find genes that protect you from disease. And the reason is, and in particular, I want to find genes that when broken, like they don't function. So the gene has its normal function, you have whatever the average rate of disease is. But I want to find genes that when you break them so they don't function at all, you don't get disease or you're protected from disease. And the reason we want to do that is because A, it's relatively easy to recognize a genetic mutation that breaks a gene, and it's very hard to figure out all the rest of them that make the gene go up and down and have subtle effects. And so someday we'll know how to do that. That's a big research program here, is to figure out how to read the genetic code and all its myriad subtleties. But it turns out today the one thing we're really good at is we can recognize when a DNA variation breaks a gene and stops its function cold by like truncating it, just chopping it in half. And the other reason we want to do that is it turns out the easiest drug to develop is an inhibitor. So in other words, if you think about drugs you might develop, some drugs will stop something from functioning, inhibit it. Others will have to replace it or augment it or change it. Oh my God. Replace, augment, and change is very hard with a drug. But stop it is actually relatively easy. So it turns out, so because of that, if we can opportunistically focus on finding genes that we can recognize have what we will refer to here as loss of function, I will refer to, loss of function, they're broken. And when they're broken, the person is healthy and actually protected from a disease, then it's relatively straight shot to say, let's build an inhibitor of that and then try that in patients, okay? So this is precedented. It's not like I'm making this up. It's not my idea. One example of this that's current is this something called PCSK9. It doesn't really matter what the gene name is. There's a gene that was first found through human genetics because there were people who had super high cholesterol levels. And it was just a rare couple of families, and they used this sort of study the whole genome approach, no hypothesis, and said, oh, there's a gene called PCSK9. No one had ever studied it before, but it seems like if you have a mutation in it, you get higher cholesterol. And then somebody realized that mutation actually wasn't one that reduced the function. It actually was so rare, it was a very rare mutation that actually turned on the gene. So somebody else said, Helen Hobbs in Dallas and Jonathan Cohen said, well, if activating it increases LDL, maybe turning it off will lower LDL. They went out, and it turns out there are relatively common changes in this gene of like 1% frequency that actually knock out its function, okay? And if you inherit one of those, your LDL cholesterol is lower, and your rate of heart attack is 50% lower. And those have proven to be very robust findings, reproducible. And it turns out that a Harvard medical student, when she was spending a year in at Helen's lab in Dallas, found an individual who lacked both copies, both from mother and father. This is very rare, but it's 1 in 100 squared is 1 in 10,000, so it's not vanishingly rare. And that's the squared thing, that's genetics, it's for tomorrow's lecture. 1 in 10,000 people or so lack it altogether, and people have studied those individuals, and they've never seen this protein, never made it, and they're perfectly healthy. The anecdote, which is a true anecdote, is the first patient they found that completely lacks PCSK9, doesn't make it, it's not detectable in their body, lacks the gene, is a college-educated aerobics instructor, mother of 2. Okay? So the idea was that if you could lower the function of this PCSK9, you would lower the LDL, lower the rates of heart attack, and it would be safe. And there's now like 14 companies that have developed agents for PCSK9, and there's an article in the New York Times you can read by Gina Colada that cites somebody as saying, the biggest worry everyone has is the LDLs are going so low, no one has ever seen an LDL that low before, although the depth to which they're being lowered is actually not as low as that woman who actually lacks the gene altogether, but this is now in phase 3 trials, and we'll see what happens. But people are quite, if you had only projects like that, it would not be a 5% success rate, okay? So can we find more? So we published a paper a few months ago, and this is the last sort of bit of my story, that again has many authors because it's another story that was hard to publish because people were skeptical, and I'll tell you why. We had to get the whole world to work together, and so we did. It was not a problem. No, I mean, literally, this study had 150,000 patients in it. And for example, This is Jason Flanick, who's my postdoc, brilliant guy who's the first author. This is Kari Stephenson, who is a good friend who runs Decode Genetics, a for-profit company in Iceland. This was David Cox, who unfortunately passed away, another good friend who worked at Pfizer. And this is Leif Group, my colleague who I worked with for 15 years in Finland. This was truly a worldwide public-private effort to get this information out. Okay? And the idea of this was in 2009, in a public-private partnership with Pfizer, and they paid for it all and let us publish everything and put everything out in the public domain, we said, can we find people who are at high risk of type 2 diabetes and have a gene that's broken that actually protects them from diabetes? There was no such thing ever shown, but that was the proposal. And they funded it, and we worked together on it. And what we did was, working with Leif, initially we went out to a population of 28,000 people in Scandinavia that he'd followed for 25 years with his colleagues, and we identified people who were, the people we had as sort of, one group was people who were in their 40s, they were lean, and they actually had type 2 diabetes. Normally, those would be your cases. Like, there are people, geneticists like stuff like that. You have no other reason to have it, and you have the disease. Those were actually our controls. What we were interested in were the people who had average age of 66, BMI of 33, which is very obese, and on an oral glucose tolerance test, were completely perfect. And what we wanted was to know what could make somebody, and a large population of people, have obesity and older age, which are really the risk factors we know about for diabetes, and many of them had family history and didn't have any diabetes, and then we try and figure out why. And so we looked at a lot of genes, and I'm not going to go through how we did it for this audience, but just say we looked at a lot of genes, looking for proteins that were broken, and we found one genetic variant, okay, in this study, that basically at position 138 of the chain of the protein was truncated. That's X, means it stopped. And we could recognize that that was likely a protein that didn't function because it was missing most of the protein, and we went to the laboratory, or actually someone else went to I didn't go to the laboratory, but Nicola Baer went to the laboratory. I haven't actually been in the laboratory, no, I'm just kidding, in a long time. When my son Zachary asked me once what I did for a living, I said I answer email. But anyway, I digress. They found the, true, but nonetheless. So we found this genetic variant which truncates the protein and in fact does abolish the function of the protein, which I'll tell you about in a minute. 1 in 1,000 people in Finland, okay? And the 1 in 1,000 people in Finland who were missing one of the 2 copies of this protein, and we looked here at 40,000 or whatever this is, 48,000 people, there were 13 cases and 56 controls, and it's not balanced, so you can't just look at the numbers. But the bottom line is the risk of having diabetes was 50% in the people who carried it. Just one second. If you had this variant, your risk of type 2 diabetes was 50% of what it was if you didn't have the variant. Yes? I didn't tell you yet, but I'm going to. I'm trying to tell the story without too much jargon. So just trust me, there's a protein, and I'll tell you about it in a minute. It turned out we knew this for like 2 years, and it turns out, I won't go into the details, that this variant is only present in Finland. It's actually not only present in Finland, it's only present in Botnia, which is a Swedish-speaking sub-isolate of Finland, which is from when Sweden used to control Finland. And now, that was not unrelated to why we were studying the problem in Botnia, because geneticists like to study populations that are homogeneous and have special histories. That's actually why we were working there. But the downside is when you find something, it's not necessarily anywhere else if it's rare. So we actually went all over Europe, or we actually called our friends all over Europe. They sent us DNA samples, and we could never find this variant again, so we were kind of stuck. But then I was visiting Kari Stephenson literally at like 10 a.m. on a Tuesday morning, and I told him this story. And this is how our field really works. Like, this is an unpublished result. You could say scientists hold everything close. I said, Kari, this really interesting variant, R138X in this gene, and we can't replicate it. And since Iceland is sort of related to Scandinavia historically, maybe you would have it. And he went and sent someone, and they looked in the computer and said, we don't have that. We never saw it in our whole population. But we have another variant. Don't worry about this mouthful. It's an even more severe destruction of the protein. And it turned out they were able to tell us that they had basically, those people had an 80% reduction, so 18% the risk of type 2 diabetes in the people who carried it as opposed to people who didn't carry it, okay? So there were now 2 different versions of this protein in 2 different populations that knocked out the protein. We showed it in the lab, both of them don't function. Okay, the protein's encoded in that way. And they were protected from type 2 diabetes, and also Kari showed that actually in people who didn't have diabetes, but who carried this, their glucoses were lower, okay? So what is the gene? my friend wants to know. And so the gene is called SLC30A8. It's a great mouthful. What is it? It was actually first found to have something to do with diabetes in what's called, I didn't introduce this slide and I'm not going to bother, but the studies that we've done and others have done to make it possible to scan the genome for genetic variations that affect disease really came online around 2007. And these are sort of the number of genes known for type 2 diabetes. And there's lots of them now. I sort of skipped over this because I was blathering on too long. But in this first wave of 2007, this gene SLC38 was found, not because of these genetic variants, but yet another genetic variant in this gene that it was unclear if it broke the gene or up or down regulated. It was a very subtle tweak in the gene and it had a small effect on diabetes, but it was strong enough to have discovered it out of the whole genome as one of now 80 genes that seem to affect diabetes in people. And I purposefully sort of skipped over this complexity because I'm trying to build an argument about how we might develop new treatments, but there's a lot of complexity. There are a lot of genes that play a role, okay? So it was a gene that we actually already knew about as playing a role in diabetes and actually having a common genetic variant. Its frequency, something like 30%. So like many of you carry the genetic variant version that has a subtle effect on your diabetes risk, like 20% in this gene. And now we found that a very rare, distinct mutation in that same gene, or two of them, can lower risk of type 2 diabetes and appear to do that by abolishing the function of the protein. And so this gets to one of the key issues for drug discovery, which is, do I want to inhibit it or activate it, right? In other words, I need to know if I'm going to develop a drug, I might need more or less. The problem in this case of this, and one other thing about, I should say, this SLC30A8 gene is we actually do know what its biology is. We actually knew from work done before the genetics was figured out, about 2 years earlier, somebody was studying the beta cell, which is the part of the body that makes insulin, which is the key thing for diabetes, and it turned out that insulin in the beta cell is packed into little balls called vesicles, complexed with zinc, okay, like zinc. And this like zinc. So this protein, SLC30A8, or encoded by the SLC38 gene, it turns out puts the zinc into those little balls, okay, that the insulin's in. And everybody assumed that the way this worked was that zinc was good, because the natural state of man is to have zinc in your vesicles, actually the natural state of mice also, which is where this work was done, is to have zinc in your vesicles with the insulin. And this protein had a mutation in it before ours, this common genetic variant, and it was sort of assumed that the way it worked was that like you were abolishing the normal function of the gene, lowering the zinc in the vesicle, and that somehow led to type 2 diabetes. That was how people assumed it worked. And on that principle, you would want to augment zinc. And in fact, a clinical trial was proposed of feeding people zinc based on this. Not a very well-intentioned clinical trial, also because, or not a very well-motivated one, because even if that were the case, it's not clear that eating more of it would, like the body has this thing where it regulates everything. So if you ate more of it, it does not stand to reason that more of it would get into these vesicles, but also we really didn't know up or down, okay? The key insight from these genetic variants is that they said that lowering the function, because it was very clear that our genetic variants were lowering the function, were protecting the disease. Counterintuitive. Why would there be a gene, the zinc, it's there in everyone. Why would it be like taking it away or reducing it would make you better? But we don't know, because we don't do hypothesis-driven science, okay? Like a lot of people think that science is hypothesis-driven. You're supposed to have an idea. I sometimes refer to hypothesis-driven science as hypothesis-limited science. Like if you have a good hypothesis, a hypothesis-driven science is a wonderful thing. But if you don't actually have the right hypothesis, you might never get there. So actually the way science works is not all hypothesis testing. It's hypothesis generating and then hypothesis testing. So the kind of work that we've been doing up until now is hypothesis generating, right? So you find a gene that plays a role, now you have a hypothesis. So these genetic studies had raised the hypothesis that SLC30A8 was causing diabetes when it was mutated, and our data turned that hypothesis on its head. And so we couldn't get the paper published. So we submitted the paper, and it was rejected twice because I was going to put in the reviews just because I do that for fun, but I actually do. I don't know who the reviewers were, so I just complain about it in every audience. So I figure if they're in the audience, I want them to know how I feel. And the way I deal with it is I don't make fun of them. I just tell the story in such a way that everyone laughs when I show the review. So, because what the review said was this data is unpublishable because everybody knows from the mouse studies, and this isn't even what the mouse studies showed, everyone knows from mouse studies that this causes diabetes. and that was unpublishable. So we had to actually go to a different journal, and we had to collect a lot more data. But luckily, in the period of time that the review was going on, we'd sequenced like 13,000 more people with diabetes, and we found 10 more mutations. I spared you the data. So we've now found 12 different mutations by looking at 150,000 people in total. And on average, if you carry any one of these 12 mutations, which is still a very rare thing, to carry any one of the 12, your risk of diabetes is about 65%. And we got the data to be so overwhelming that they just said, okay, we give up. And now it's accepted that it's the case that you actually want to inhibit this thing, not activate it. And the biotech company that had been started to activate it had to switch gears, okay? Because there was a biotech company founded to activate it. But actually, that's okay, because if you're a biotech company, you're activating it, it's much easier to inhibit it. You're probably not unhappy, you're probably happy, because you can go do that. So it turns out that this piece of work says SLC38, you might want to inhibit it, it might protect type 2 diabetes. Is this the only one? I told you about PCSK9, but we actually set out 5 years ago to find more things like this. And the objection was that if they were knowable, they'd already be known. That often is what happens when you propose things, and so it was actually hard. I'm gonna have to stop in a second, but I'll just note that Sate Kathreson, who you met earlier, just published a paper in the New England Journal about a month ago where he published another gene, ApoC3, that it turns out reduces risk of heart attack. It has 4 different loss of function mutations in it, and it turns out if you carry them, you have a lower risk of heart attack. And this acts through something called triglycerides that if you read the textbooks, says triglycerides have nothing to do with heart attack because they're correlated with HDL, and HDL is the thing that has something to do with heart attack. And what Sake's work has rewritten the textbook says actually the HDL may be the innocent bystander of triglycerides, okay? But this is a specific target, ApoC3, and it turns out that a biotech company had already built a way to inhibit this, so they now can see if that will prevent heart attacks. So with that, and we actually have found a couple of others as well. So I'm gonna stop and just say, the goal of all this that I've described, it's only one of the ways you can use genetics, and it's only one of the questions you could ask about diabetes and genetics is, how are we gonna figure out what we'd wanna do to prevent or treat the disease, and using experiments of nature to try and figure out what might be most likely to work so we don't develop drugs that work in a mouse and don't work in a person. And if you could do this across all of disease, I think it would help with those success rates and might lead in the long run, but it'll be a long road to better treatments for patients. So with that, I will stop, and that's my backyard and my lab and my colleagues. So thank you very much. Do you have a question, sir?
I'm confused about the issue of whether type 2 diabetes can be reversed, cured, because I've heard different views on that subject. Thank you.
Sure. So the question is, can type 2 diabetes be reversed or cured? If you take all comers, so in other words, you take lots of people with type 2 diabetes and you look at clinical trials, randomized trials, which are the only way really to know, okay? There's never been a treatment shown that will, remember that curve I showed of the glucose going up with time or the diabetes rate going up with time? There's never been a therapy shown that just stops that curve or that reverses it. However, embedded within that population are no doubt some people who get better and some people who just continue for whom that treatment had no effect. So when I was taking care of patients, I definitely had patients who would come in, they had diabetes, they would change their lifestyle, and they'd be all better and it would just be stable for their life. But of course I had other patients you'd put on every medicine known to man and their diabetes would get worse and worse. And so we have no ability right now to say who's who. All we can say is on average, the therapies we have do delay the disease by a couple of years, but they don't eat for each new pill or treatment you add, but they don't stop it on average. So for you or for someone else, it may well work, but for everyone it works in, there must be someone for whom it doesn't work at all, because on average, it doesn't work very well. Yes?
Related to that question, you said diabetes is growing dramatically. It's related to lifestyle,
yes.
Obviously also related to genetics, but that lifestyle changes don't work overall. They don't work enough, the curve still goes up. So is it that lifestyle changes aren't dramatic enough given the environment that we're in? Or, I mean, is that part of what the question is?
The analogy I like to use, it's a great question, and just to be clear, to parsecs, it's sort of a subtle distinction. There's no doubt that the changes in the environment in which we live are the cause of this explosion of diabetes. It stands to reason, and I believe it's the case, that if we went back to living the way we lived 100 years ago, that would go back down. But it does not stand to reason that an individual living in that society is able to make that change on their own. And the analogy I like to use is to other types of reward behaviors or addictive behaviors, because I think eating is sort of like that, like smoking and drinking. If you think about it, individual smoking cessation was very imperfect and unsuccessful, but societal change, so that in America at least, the sort of demonizing smoking, no smoking in restaurants and public places, has had a major effect and a positive effect. I think that I'm all in favor of, and actually quite optimistic, that if we had public policy, public health, societal changes that led to a healthier lifestyle, it would in fact stem this tide. But the analogy to smoking is, in a room, in a bar where everyone's smoking, it's extraordinarily difficult for an individual to quit. The other analogy is, you'll observe that no one quits smoking by going from a pack a day to 2 cigarettes a day. No one quits drinking by going from a bottle of vodka a day to a shot of vodka a day. You have to go cold turkey. And that's just something about the way our minds work. You have no examples you can give of addictive behaviors that you sort of ratchet back. The trouble is you have to eat, right? No, I'm serious. I'm serious. So so the trouble is, show me another example of a behavior that has it's the same system in the brain, where you sort of say, there's all this great stuff around, and I'm gonna go back I'm gonna go to 50% of it. There's no example of that in the other analogies. So I think that it is a societal change would no doubt work. I see no evidence that we're gonna do that, but the people who do that, I think, will probably have bigger impact if they could ever achieve it than we will with our treatments. But I still think even if they did that, it would only be 150 million people. Right, so there's still I actually do get the question sometimes very skeptically, why do you people take all the resources for genetics when we should be doing all the important environment and public health stuff? And that is a false dichotomy. There are people who have the disease, and it is a good thing to try and figure out how to actually help them. And then there are other people who should change and do these other things. Yes?
Hi, doctor. First of all, phenomenal talk. And my question is, I know in Sake's paper on ApoC3, that's an exome-wide association study.
Yep.
Do you think there's any value in studying the introns of the non-coding sections of DNA?
So there's the question, which is a little bit of inside poker, but important question is, how can we most effectively go about these studies? And it really has to do, and how much of the genome do we study, and how do we deploy our resources? There's a lot of debate in the field. We're better off doing more people for focused parts of the genome where we believe we know how to interpret it, or are we better off doing fewer people more completely? And I would say the real question is, what's your question? And not your question, but what's the thing we're trying to do? If you want to, in the short run, discover things that today we can interpret, sequencing the parts of the genome that encode proteins, which is the 1% called the exome, is the most interpretable. So if you want to know that, that's the most efficient thing to do. It turns out you can do that more cheaply and rapidly, et cetera. But it turns out that of all one of the things we've learned from genomics that I actually think is perhaps the most profound thing we've learned from genomics is that when I was a student in college, I was taught, and everybody was taught when I was in medical school, that the genome consisted of the parts that encode the protein, that was the functional parts, and then what's called the junk, which was the 99% that had no function. What the genome taught, and actually there's a great editorial in Nature against the Genome Project in 1986. I could show you it if you want, because I carry it on my laptop, because I think it's funny, and it says, some people say we should sequence the genome to learn about the functions of bits outside of protein coding genes. That might be valuable, but you're certainly not going to learn about that by sequencing the genome. And it turned out the only way we learned about it was by sequencing the genome and methods derived from that to actually have the sequences. And it turns out we now know, a lot of this work was done at the Broad as part of international consortia, that yes, the 1% that encodes proteins is functional, but it probably makes up about 10 to 15% of the functional DNA in our genome. So our best estimate today is 90% of it probably is junk, as best we can tell with today's methods, but there's like 10% or so that's functional, and or maybe 8% that's functional, and 1% or 1.5% encodes proteins, and the other, let's say 6.5%, does everything else that we don't really know. Because it's that geneticist approach. We now know it does stuff, we don't know what it is. So if you ever want to understand that, you're going to have to sequence the whole genome and figure it all out, but that's going to take 10 or 20 years. If you want to figure something out next year, you can focus on the proteins, but if we only do that, we'll never understand most of how our genomes function. So probably we should stop. Thank you all for coming. Okay. It's great. I really enjoyed being here.
Yeah, thank you all for being here. Thank you to David again.